Hello, my name is Miley Burke, and I work in the Morrison Lab under the mentorship of Dylan England and Dr. Morrison. I will be discussing our research this summer on deciphering the metabolic pathways of the INO80 Chromatin remodeling complex by genetic analysis of Saccharomyces cerevisiae. In order for cells to thrive in different nutrient environments, they must regulate their metabolism. They can do so via changes in gene expression, a process that is regulated by remodeling chromatin. This facilitates a wide variety of crucial cellular processes, such as transcription, DNA reparation, and DNA replication. One way that chromatin can be remodeled is by chromatin remodeling complexes like INO80, a structure that is essential to metabolic stability. Thus, the disruption of INO80's activity can result in disease and cancer. In fact, recent studies have found INO80 subunits to be amplified in as much as 65% of xenografts from breast cancer patients. This research is thus important to better understanding INO80's activity and how the specific disruption of which can contribute to cancer. With this, technology like CRISPR might be used in the future to precisely fix these disruptions in INO80's cellular pathway and prevent diseases like cancer. As INO80 and its subunits are evolutionarily conserved, we can use the yeast Saccharomyces cerevisiae to better understand INO80's cellular activities in humans. Two subunits at the enzymic center of INO80 are Arp5 and Ies6, the deletion of which has been found to disrupt INO80's activity and the metabolic stability of cells. However, precisely how these subunits coordinate their activities within cellular pathways to remodel chromatin is currently not well understood. Thus, we sought to study the genetic interactions of 4 genes in the cellular pathway of INO80: NUM1, MIP1, SLA1, and VRP1. With its Arp5 and Ies6 subunits to better understand the mechanisms by which Arp5 and Ies6 regulate the activity of the INO80 complex. These genes were chosen in particular for their involvement in mitochondrial function, as well as metabolic signaling. As a secondary project, we also developed a tool for future genetic analyses. Ies6 displays prion-like behavior when overexpressed, and prions are essentially intrinsically disordered proteins that can drive the inheritance of new traits. These traits arising from prions are conserved from yeast to man, so inducing the overexpression of Ies6 could be a useful tool for the switch-like induction and loss of heritable traits. My project sought to create double mutant strains of Ies6 and Arp5 with the genes NUM1, MIP1, SLA1, and VRP1 to identify the genetic pathways in which these genes are involved. The first step in doing so is to create a linear piece of DNA fragment lacking the desired gene. We did this using fusion PCR, knockout primers, and gel electrophoresis to then visualize the DNA links. Knockout primers work by amplifying a selection marker from a plasmid. It has flanking sequences to direct it to recombine in place of the target gene. The selection marker we used was a G418 antibiotic resistance gene called CAN. We then transformed these genes into yeast shuffle strains. These shuffle strains have chromosomal Ies6 and Arp5 deleted, but they also have a plasmid containing a wild-type copy to prevent the temporary sickness of the yeast due to the deletion of Ies6 and Arp5. We then grew the yeast on G418 YPD plates to confirm that the gene was indeed knocked out. If the yeast were able to grow on the antibiotic plate, that confirmed that the gene was indeed knocked out. We then used colony PCR and gel electrophoresis to further confirm positive results in which the genes were successfully deleted. We then grew the yeast on either -5 FOA plates to lose the Ies6 or Arp5 plasmid, or on -0 plates to retain the plasmid. This provided us with wild-type, single deletion, and double gene deletion yeast strains. As for our secondary project, we created a tetracycline-inducible Ies6 overexpression system. As for our results, we attempted to create double gene knockouts of NUM1 and MIP1 with Ies6 and Arp5 using 2 opposing transformation methods. The first method we used was to delete NUM1 and MIP1 first, and then Ies6 and Arp5 second, and the second method we used was the opposite, where we deleted Ies6 or Arp5 first. However, we found the yeast to be highly resistant to this mutation that we were trying to induce, giving rise to only 1 successful double MIP1 Arp5 gene deletion among 3 transformations and 41 individual yeast colony screenings. We thus concluded that perhaps the combination of these genes is lethal in Saccharomyces cerevisiae. However, to confirm this result, we will proceed with an alternative transformation method using heterozygous NUM1 and MIP1 instead. As for our other genes, we successfully created double mutants of SLA1 and VRP1 with Ies6 and Arp5. We are currently in the process of conducting fitness assays to compare the cell proliferation of double gene deletions with the single gene deletions and wild type. The combined effect of these two gene deletions upon yeast fitness will inform us whether these genes function in the same, parallel, or closed pathways. Because INO80 is evolutionarily conserved, a fitness defect in yeast might well translate to a developmental defect or disease in humans. Lastly, we found that the drug doxycycline, which is a tetracycline analog, induced prion-like behavior in IES6 as there was high replicate variability for IES6 with only the prion-like domain, which is a common trait of prions. However, there was surprisingly also high replicate variability for IES6 lacking the prion-like domain. Thus, perhaps the overexpression of IES6 without the prion-like domain disrupts wild-type IES6 prion function. However, we are going to proceed with more experimentation to better understand this unexpected result. Thank you for listening.